In the previous videos, we talked about peptide preservation and dissolution. Many interested friends have left messages in the background, hoping to learn how Qiangyao Biotechnology synthesizes peptides. Therefore, today we will talk about the principles and steps of peptide synthesis. The peptide synthesis of Qiangyao Bio adopts the solid-phase peptide synthesis method. The basic principle is to continuously add, react, synthesize, and operate amino acids in a known order to obtain the desired peptide. Its biggest feature is that there is no need to purify intermediate products, and the synthesis process can be carried out continuously. In order to facilitate everyone's detailed understanding of the principle of solid-phase peptide synthesis, we will introduce the specific operation steps manually below. 1. Hanging resin. When using, take a certain amount of resin and add an appropriate amount of DCM to a clean and dry reaction column for swelling for 30 minutes. Remove the solvent and then add the first amino acid with FMOC protection, DCM, and DIEA for 50 to 60 minutes of reaction. Remove the reaction solvent and wash twice with DCM. Add an appropriate amount of blocking solution for 20 minutes. Remove the solvent and wash twice with an appropriate amount of DMF. 2. Remove FMOC protecting group. After the resin is hung, add an appropriate amount of 20% piperidine, DMF, for deprotection twice, each for 10 minutes. Wash 6 times with an appropriate amount of DMF. Three. Testing. Take more than 10 pieces of FOAP, removed resin, and place them in a detection tube. Add 2 drops of Kaiser reagent each, and place them in a dry, wet incubator. Heat at 105 degrees centigrade for 3 minutes until the resin turns dark blue. 4. Amino acid linkage. Weigh 3 times the amount of amino acids, HOBT and HBTU, and add them to the reaction column. Then add an appropriate amount of DMF. React with 3 times the amount of DIEA for 30 minutes. Remove the reaction solution and wash 2 to 3 times with an appropriate amount of DMF. Take more than 10 pieces of the reacted resin and place them in a detection tube. Add 2 drops each of Kaiser reagent and place them in a dry, wet incubator at 105 degrees centigrade for 3 minutes. Resin colorless indicates complete reaction. Repeat the above steps until the peptide sequence is complete. Then remove the FMOC protecting group on the last amino acid to complete the synthesis of the entire peptide sequence. Wash 6 times with DMF. Then wash 2 to 3 times with an appropriate amount of methanol. And finally vacuum dry and weigh the tube. At this point, the entire peptide synthesis process is completed. In general, it is to add resin, then connect the first amino acid to the resin, deprotect, and add the next amino acid for reaction. and then deprotect and add new amino acids. Repeat the above steps until the peptide we need is synthesized. Friends who are interested and have the conditions can try it out themselves. If you have any questions during the experiment, you can contact our online customer service, and we will be dedicated to serving you.